So first of all, thank you so much for inviting me to present here. And secondly, thank you for being here today with me to at least share our very exciting story on what we are currently doing with HiFi sequencing to assess how we can use them in a clinical genetic setting. But first of all, I would like to share with you that, of course, although I'm here with you in this PacBio event, PacBio is heavily involved in what we try to achieve simply by sponsoring our studies. But they have not been involved in the conclusions, nor the design, nor the results that are derived from this study. So although I'm here to share with you that HiFi sequencing is absolutely wonderful and they all should use it, this is not something PacBio is paying me to say, because this is something that we truly believe in from a clinical perspective. As I'm not quite sure where you're all coming from and what it is that you do, I'd like to start relatively easily by sharing with you that I work on rare diseases in a clinical genetic setting. And there are roughly 7,000 different rare disorders. 1 in 17 of all of us will have a rare genetic disorder manifesting somewhere throughout life. The vast majority of them will have a genetic cause. Currently, 30% can be diagnosed. 75% of all these genetic rare disorders actually already manifest during childhood. 30% of all patients die from a genetic disease early in life, before the age of 5. And unfortunately, it currently takes 6 years to get the right diagnosis on average. And to do this, not only they see many different medical specialists, which all, of course, are very excellent in their own medical specialty, but relatively not always look at the entirety of the human patient or of the patients as a whole to diagnose this rare disorder, which makes them see sometimes 12 different medical specialists. And very sadly, 1 in 3 also receives a wrong diagnosis, which, of course, if you think from a clinical perspective, has its own track, trying to prevent further misdo in clinical practice, or at least prevent further harm to be done. So what we hope for is that by studying these rare disorders with the best genetic technologies, that we can not only find the genetic cause easier, but also more accurately and faster, and that helps them patients not to have the diagnosis at the end point of their genetic care track, but actually make it a starting point to further develop therapies and impact their clinical management. This is just a summary slide on where we have come from rare disorders from a genetic point of view. If you would take the pre-next generation sequencing era, approximately 7% of patients would get a diagnosis. Over time, we have followed several cohorts of over 150 patients each to see how technology impacted the way on how genetic diagnosis would be identified. And initially, we followed this cohort where 7% actually got a diagnosis by using exome sequencing. At the same time, you can show that 1 in 3 actually can get a diagnosis by simply replacing one genetic test for another, allowing a better unbiased way to look at a person's genome. We also know that if we do the same thing comparing short-read exome sequencing to short-read genome sequencing, there is some increase in diagnostic yield, but not statistically more. And the most reason to actually find more is that you can better detect structural variation, but the limited increase in diagnostic yield is because we, of course, all expect that some of these mutations or these pathogenic variants are located in non-coding space, where we currently lack the tools in clinical genetic testing to really prove the pathogenicity of them. The highest increase in diagnostic yield today, if you follow again one of those cohorts in time, is actually reanalysis of the existing data, simply because new information becomes available, especially working in rare diseases. There's still new disease genes being discovered, I would almost say on a daily basis. That's no longer the case, but at least, every week there will still be new disease genes discovered. And if you reanalyze existing data, you can actually increase, if there is a true genetic cause in a very well clinically pre-selected cohort, that you can increase the diagnostic yield to 50%. But still 50% is not having a diagnosis. And of course, this is where we believe that long reads may help. So one of the things that we do in our clinical genetics center, and I'll show you where I come from in the next slide, is that we want to impact this diagnostic track by moving not only the genetic test to a first test if genetic testing is being done, but also to have it the most comprehensive analysis in order to make the fastest diagnosis possible and assessing the real human genome whenever needed. At this moment, it may also be no surprise that exome sequencing in this context might still be the best cost-effective way to look at the genome. Yet, if we do reanalysis over and over and get to learn more on how to interpret non-coding space, of course, you may also wonder whether that might still be the case in 5 years' time or so. And then, of course, it becomes the question: if you want to use if you want to use a genome because you believe that there might be more to find to solve that 50%, what genome do you then need? Do you then go for a short-read genome, or do you go for a long-read genome? And our mission in our diagnostic hospital, or at least in our diagnostic department, is that we work a lot together with another university in Nijmegen, in the Netherlands, which is called Maastricht. So this is where I'm coming from in the Netherlands, and this is our partner affiliation, where we combinedly care for over 2 million people, genetically speaking, for the diagnosis. We have in our department over 25 different PI groups working on rare disease genetics, from very fundamental understanding up until therapy development. And our diagnostic setting detects, or at least services, more than 40,000 patients annually. So if you take this into account and want to see whether you want to go for short-read genomes or long-read genomes, we also have to take into account the volume. If you want to go for a one-test-fits-all, so you have one patient coming in and they all get the same mainstream type of testing, we need at least to be able to service our community, having a throughput of nearly 40,000 samples a year, but also to have a turnaround time of a test that is suitable to impact their medical decision making. So it can't be 6 months before a patient comes in and receives a diagnosis, but preferably we will be able to do this within a week or so. So this is where we want to move to. Service all these patients in one go. For negative cases, move them into our research section of our departments to make sure that also their further developments are ongoing to show pathogenicity of certain variants. So in our aim to look for a one-test-fits-all strategy, it may also be no surprise that after going from exomes, we started to see whether we could actually replace all of our postnatal germline diagnostic testing by short-read genomes. Because also there seemed to be an incentive. Long-read HiFi sequencing was not as available as it is today when we started this study. So what we did is that we took an overview of what we were doing in 2022 on germline genetic testing across our center together with the one in Maastricht. And what we noticed is that for germline testing, we did roughly 25,000 patients. All of those are referred to our clinical genetics center, and all of these patients, when diagnostic testing is being requested, together these 25,000 patients lead to more than 30,000 genetic tests. And that is a clinical referral that says, I believe this patient may have CHARGE syndrome, and then in the lab this is being translated to what experiments need to be performed. And CHARGE syndrome is a rare syndrome caused by de novo mutations in a gene called CHG7, and this can be either a point mutation or a small insertion deletion event, but also copy number variations can lead to this disease, making multiple workflows to be used or multiple type of assays to be used in order to find, if present, the relevant genetic variants. So 25,000 individuals lead to over 30,000 or 36,000 experiments to be done in the lab to serve these patients. Whereas if one test fits all would work, you would essentially be done with 25,000 tests, one per patient. So that would usually reduce our complexity of our laboratory workflow. We would not have to maintain all these different workflows to work with, but we could only use one efficient genome sequencing workflow. So we started with our experimental setup. What we did initially is that we selected of all of our positive cases, so they had a known genetic cause of those 22,000 of the cases seen in 2022, and we took 1,000 samples that are representative for our cohort of genetic testing. We took the ones from targeted analysis, we had genome-wide analysis, so exomes, karyotyping, etc., etc. We classified them according to what type of assay was used initially, and the types of variants that we were initially identified in the original track to see whether we could also identify those pathogenic variants in short-read genomes. What we noticed is that we could detect a lot, but not everything. And that if we were to look for a one-test-fits-all strategy where we would actually phase out all of the other workflows and focus only on genome sequencing, that here in the darker pits of these pie charts, that is the amount of reduction that we could achieve in a certain workflow. You can see that none of these circles, apart from the exomes, is fully replaceable by a short-read genome. Some are logic, I would almost say, because some of the tests, of course, are used for repeat expansion analysis. We also know that that's not as easy in a short-read genome as one may expect. But some of the other assays, like, for instance, karyotyping, we could not replace. That is not because you can't detect structural variation in a short-read genome, but because in our hands, we use a lot of our karyotyping to find variants that are of somatic origin. And, of course, doing a 30x short-read genome, that will not be replaced by you can't replace karyotyping in this particular reason for testing by a short-read genome. So very disappointing to our initial setup, because we of course had hoped that we would replace everything that we were doing by a genome, but we couldn't. If we translate this back to what we can replace approximately 80% of all our clinical indications by a short-read genome. But all of these other 133, so roughly 20%, we cannot. And the reasons why we can't replace them have to do with the typical mutation detection that is not apprehensible from a short-read genome. But if we look at patient level, that is 25% of all of our clinical referrals. So short-read genomes are great, but they are not our one-test-fits-all strategy. And of course, I'm here because we believe in long reads, and I'm going to show you why these are better. Short reads are great, long reads are better. For a long time, we have already been using long read-based amplicon strategies. What we have been doing before is that we have quite a few genes in our clinic, and they are listed here on top, for which we know that there's pseudogenes or homologous regions in our genome that we can't use an efficient PCR-based assay or an exome or a short-read genome or any other short-read assay that is targetable. So by picking primers that are uniquely positioned at the long-range fragment and then using those on a long-read sequencing platform, we were actually able to already use this long-read sequencing, but in a very targeted way for a single gene each, and sometimes multiple amplicons still to capture a single gene. But we have been using this very efficiently from 2021 onwards on the PacBio SQL system to just simply allow an easier test. It's not one test fits all, but an easier test than what we've done before. Something else that we've been looking into is what are then the regions, if you would do a long-read genome, that you would capture, and how relevant are these if you compare it to short-read genomes. If we look here, this is just a schematic ideogram of the chromosomes, and all of these red boxes are locations in our genomes that we could not capture using a short-read genome, but that we were able to capture using a long-read genome. And this is, of course, the experiments being done on the same set of individuals, and then directly comparing the outcome of each of them. So in our hands, we could capture 35 megabases of sequence only using the long-read system and not by short-read genomes. The question, of course, is how relevant are these regions? In our clinical setting, do we then get better results? Are we better servicing our patients? If you look at all the variants, 2% in these regions is actually it's coding sequence. So already directly interpretable in a clinical genetic testing setting. Of these, also 64 genes are reported in those, or at least the variants are reported in these 64 genes. We know that 50% of these genes are already known to be involved in rare genetic disorders. So although we have not been able to detect any new pathogenic variants in these regions in the patients that we have tested in this approach, it's of course also only a matter of time before we actually do. Another important note is it's not a single type of disorder that you then service better, but we also, across the whole spectrum of rare disorders, are now capturing variation that we couldn't otherwise detect. One of our latest studies that we have been doing using the HiFi genomes is, again, comparing the accuracy and sensitivity to actually detect de novo mutations in HiFi genomes. And what we've seen here, and this is again the same set of patients run on short-read genome sequencing and HiFi long-read genome sequencing, allowing a direct comparison on the variants that are detected. And what we see is that you actually capture a large fraction of new variation as expected that you would only capture using the long-read genomes. We have here used 8 trios with neurodevelopmental disorders, and the variant data calling sets are available online if you like to for further comparisons. But our main conclusions is that there is a strong concordance in what you can call on both of the platforms. You win some, you lose some. This is always moving to a new technology, if you like. But our main gain in using the long-read genomes is, of course, the structural variant detection that are mainly uniquely identified in the long-read genomes. And because we have been, well, at least for neurodevelopmental disorders, have been so well primed that those structural variations are a prime cause of this type of disorders, we believe that looking for long-read genomes in these kind of patient sets is actually quite valuable to move further onwards. So this is also the reason that we in our technology center have invested heavily in all of the different equipments. We still have our short-read genome sequencers, but we now have also a large fleet of our long-read genome sequencers. But the question is, are we there yet? What can we detect? Can we detect all of the relevant things if you would repeat our 1,000 genome studies that we did with short reads, if you repeat them with long reads? Can we then also do what we expected to be doing? So this is where we go for our Revio 500 study that we have been doing together with PacBio over the last year, and we're wrapping up as we speak. And this is a study where we try to uncover the genetic origins of rare diseases and to see whether we can use and replace our current genetic testing, the 1-by-1 version, one test replaced by long-read genomes, to see whether you can capture the variants that you expect. We have divided our studies in 3 tiers. So the first tier is testing 100 samples with known genetic aberrations that are challenging to detect in short-read genomes. And the second 2 sets are specifically chosen to see where we can improve diagnostic yield. So for these patients, there's currently no genetic diagnosis, despite sometimes having a clear phenotype suggestive of being genetic, but also to have one, for instance, pathogenic variant in a recessive disease where the second allele or the second pathogenic variant on the second allele has not been uncovered as of yet. So I used the first introduction on short-read genomes, not because it's also easy for me to get going and to get you all on the same page on what we're doing, but also to reintroduce this slide, because I showed you what we could not detect on short-read genome sequencing from a technological point of view, from a variant perspective point of view. And from this, what we could not detect in short-read genomes, we have now selected 100 cases of the 1,000 that we had done before and we could not detect to see whether we can actually detect them using the long-read genomes. This is a sort of easier breakdown of what we are then doing. We have taken 16 different loci on homologous or pseudogene loci. We have several different loci for repeat regions. We have the pseudo-autosomal regions included. We have polypolymer repeats and stretches and variants in those. You have the more complex structural variations that you, if you know what to look for, possibly could be identifying. We have imprinted and methylation defects and some of the other things where we believe that there is an added value of long-read genome sequencing if you would have this all in one genetic diagnostic test. So, how good can we do? This is one of the easier examples. We could not relatively easily detect this in a short-read genome, but this is an exactly determined 3,243 base pair deletion in the SMAD3 gene. And the reads here are visualized based on the haplotype colors. And here you can relatively easily see where this deletion is present. This is in a relative repeat rich region, so this is why you can't detect this with short-read genomes. But it's clearly visible. You don't need to do an awful lot of massaging of your data. This is just routinely analyzing your data, plotting and visualizing it in IGV. So this is the easy stuff. Another easy thing is this is the insertion of a mobile element, just opposite of, at least in the start of the NF1 gene coding, another specific rare disease. And again, colored by haplotype, you can relatively easily see that there's something going on here which we could not detect in the short reads, but are easily identified as a mobile element insertion in a long-read genome. Something else, a methylation defect. We already heard about with the introduction speech that there's also a value in looking at the methylation right from the same experiment and having this information available for interpretation. This is a case with Temple syndrome, which is a methylation defect of the MAC3 gene, and usually this is caused by the maternal segmental UPD. So, but in this particular patient, and here we've plotted the patient and both the parents, you can see that there's an unmethylation on both of the alleles, both the parental and the maternal and the paternal one, explaining in this case the de novo event of this particular case. And that had not been identified using any of the other technologies before. This is where we believed for our lab the main advantage would be to have a easygoing assay for the repeat expansions. One test, not a lot of plots, just a single genome and having all the repeat expansions right at your doorstep. This is one for myotonic dystrophy where there's a repeat expansion in the classical test that we use in our diagnostic lab of over 75 repeats. And here in this plot, you can see plotted by the haplotypes that there is something going on this particular allele with the expansion. And there's this wonderful tool from the PacBio team called Target, where you can also directly get the sequence context of this repeat. And of course, in our initial classical way of looking at the repeat expansions, this information was missing. But you can also imagine that for some of these diseases, the actual sequence context itself matters. You get the information on the interruptions, you get information on which type of sequence is actually amplified or at least expanded. And here you can see that it's also, again, that it is the relevant repeat that is being expanded and explaining the phenotype in this patient. So there's a lot of advantages of also, again, having the long reads at the phases, but also at the exact molecular level, allowing you to determine how many repeats and also the sequence context. You also get an exact estimate of the repeat length. And I think that what we can now also get into is to see how well and how broad the spectrum of all of these repeats really are, rather than saying it's above the pathogenic repeat size, but not having the accurate level of information. This is the overview of the samples that we've been doing so far. We have not fully completed our studies as of yet, but you can see that this is the relative number of success rates of the samples that we've tested in this category. And overall, I would say this looks great. There's just a few red boxes that I would like to highlight to you, because if I were you, I would wonder what that is. There is an overrepresentation in our hands, because this is what we see a lot in our clinical practice, of RFC1 repeats. So we had included a couple of them in this study, and 3 of them failed. And I'll explain to you in a minute why. There is also 2 repeat-mediated translocations that we have so far missed, simply because the breakpoints are within repeats that are even larger than the extent of a long read. We, of course, knew that would be challenging, but we did not want to spoil, or at least not be representative of what we were testing. So we also included these to see how well or how bad, depending on how you want to look at it, but where the possible improvements might be to also detect these later on. So for the first part, we believe that this has to do with the G quadruplex formation. And if the expert eye may have already seen that the ones that we miss all relate to GA repeats. And this, creates a sort of steric hindrance in actually doing the good sequencing that HiFi sequencing offers. So we believe that there might be an actual way to do better and to improve on these. The others have to do with the repeat repeat-mediated translocations. And this is where we believe that we may actually benefit from looking at other genome references, also possibly making a de novo assembly, which we currently have not tested as of yet. Just to give you one more flavor on one of those failed repeat expansions amongst this data set of 100 cases, this is a standard HiFi sequencing where you just have no coverage on this locus given the difficulty in sequencing this GA repeat length. There is now a new software release that sort of rescues some of the reads that have not fully passed as a HiFi reads, but actually do contain the sequence context. And then you see that there is some improvements, but again, not on all the levels that we can detect this variant. That it is actually rescuable is something that we have already seen by testing this pure target repeat expansion panel, where again we have included the same type of samples that failed the genome-wide level, but because of the enrichment and the higher sequencing depth that you then get on these specific loci, you can now detect very well the repeat length and the extension, if you like. So it's not a technical issue, it is simply a matter of coverage, at least for this loci that we are looking into with this GA track. We are not the only ones in the Netherlands and in Europe that are doing this. There's also a wonderful consortium ongoing, which is called HiFi Souls, and there's the different tracks involved, and I'm of course involved in the one in Europe. And we have the goal to really extensively more study those repeat and the homology regions and the pseudogene challenges. And we have selected a couple of loci across with all of these partners in Europe to see whether we can use these HiFi genomes specifically targeting a specific variant type, whereas other parts of the world are studying other types of variation that I've slightly given you a hint upon in the first 100 that we've done, but where they more extensively are studying those loci too. This is just to show you one of those regions. This is the routine exome of a patient with a mutation in this particular gene. And if you look at the which is of course terrible to interpret, you can't really make much sense of this. But if you do a HiFi genome, you can relatively very well and easily see that the variant is located where it should be, and that this is the pathogenic variant that the patient that explains the patient's phenotype. This is one of the other loci. This is the STRC locus, where again the general BAM file here is located from HiFi reads. But if you use it with homologous regions, but if you use the paraphase tool that allows you very well to distinguish the different alleles, then you can easily see that there is something going on this locus where the variant is located, explaining the patient's phenotype. So there's also very well thought through bioinformatic tricks to help us analyze our data. I again want to highlight the studies on neurodevelopmental disorders and congenital malformations, because these to us also prove the highest sensitivity of the technology, because the vast majority of these type of cases are caused by de novo mutations. And of course, de novo mutations are great in the context if you would like to study neurodevelopmental disorders, as I do. But from a technological point of view, they're actually quite challenging, because a de novo mutation also resembles a false positive event if there is a technical sequencing problem. So the number of de novo events, or so-called de novo events, or assumingly de novo events in a patient-parent trio gives you an estimate on how accurate your sequencing technology works, because you expect roughly 80 to 100 de novo single nucleotide variants in an individual that we know from other studies so far on short-read genomes. So if you would now all of a sudden detect 600, you know that there must be something wrong with your technology, or at least that your technology is not very sensitive as of yet. For the other types of variations, structure variation is expected to be 0.2 between 2 per genome, and for all of the other variant types so far understudied, we do not really have any clue on how many there are as of yet. So what we do is we select a lot of these kind of studies of patient-parent trios that have reached the end goal of that diagnostic track. So far unsolved, short-read genomes negative. Let's see if long-read genomes are better. So what we've done is we had a very strong phenotypic selection of only those cases where we believe that it must have a genetic cause. So there's lots of malformations. There is the first in the family, so all of the family structures suggest that there needs to be a genetic cause involved. And I want to show you a couple of examples. And this is the first one where this is a patient with dysmorphic features, mild to moderate intellectual disability and psychological issues. And right away from the first sequencing that we've done, so we've so far only completed, I think 7 or 8 trios. This is the first one. And right away from the variant calls, we saw 4 de novo B and D calls, or the so-called break-end difficulties, across 2 different chromosomes. So we looked further into these, and we directly could detect that this is actually a deletion insertion event on this particular chromosome here, where there is also clearly something going on the other chromosome, where there's a duplication inserted on chromosome 2. And you can clearly see that there's a drop in coverage here showing the deletion event. There is an increase in coverage for the chromosome 2 locus, where we can see the break ends that actually moved this whole segment into the other chromosome there. This is something that is relatively small. It looks huge, but it's still relatively small. And of course, this is what we were after. So if we now go into the gene context of these, we can also determine that it's a 19 kb coding sequence in NM23 on chromosome 2. And that is partially replaced by 90 kb of sequence in chromosome 3, So what we then typically do is try to reason why we have now identified it and why we did not identify it before. And for this patient, we only had an exome, unfortunately. And with the retrospective knowledge that this is going on, with the insight of what we learned from the long-read genomes, we can see that there is something going on in the exome. But I don't want to overestimate what you see here, because if you go to 70 different other loci in the genome, or 700 different other loci in the genome, your exome will also look like this, because there's a lot of deviation in copy number analysis from exome sequencing data. So you can detect it, but it's by far not sensitive enough to actually call this event without the knowledge that we now have. Similarly, for this locus here, there is only one probe that is supposedly duplicated in the exome. But again, by no means you can detect this randomly from a short-read exome, a short-read genome. So we now believe that if we look further into the biology of these genes, that you can actually conclude that this is actually very likely to be involved in the patient's phenotype, although we still lack the full genotype-phenotype correlation in the absence of a currently known involvement of these genes into disease. They're not all that difficult. This is a relatively easy case. This is a trio again, the patient, the mother and the father, where there's a single exon deletion in the FOXP1 gene, which is a known disease gene for neurodevelopmental disorders, and the patient's phenotype perfectly fits this disorder. This is also one of your questions, I'm assuming. Why did you not pick up this one before? Again, for the same reason that exomes are not sensitive enough for single exon deletions. If you have to have a genome-wide analysis, and for neurodevelopmental disorders alone, we have to evaluate 1,800 to 2,000 different genes, so it's not sensitive enough. And as we have not transitioned to short-read genomes for all of our patients as of yet, this patient did not have had a short-read genome. But you may argue that this might have also been picked up by a short-read genome in the end. To prove that this is not just our, again, local experience, we're also involved in a European project called SolveRD, where we have selected many different cases to see whether we can actually increase diagnostic yield as a whole. We have collected over 25,000 cases together across Europe with all sorts of rare disorders again. And we have had the fortunate experiment to also use HiFi genomes in these kind of disorders. So far today, we have had the analysis of nearly 300 samples across over 100 undiagnosed families, and also in these together, collectively, all joining together for the analysis of these kind of data in what we call solvatons, where we try to increase also and experience how to analyze long-read genomes altogether. We also found some more variants that we believe might actually be the cause of disease, but where we, again, lack some segregation analysis or further studies to prove the full pathogenicity of these mutations. So it might be also 15% that we can increase diagnostic yields. So in summary, we come a long way. We've done exomes, we've had a great 10, 15 years of next generation sequencing, and I think what we now have is a perfect genome. The HiFi genomes are of such high quality that you can actually look at de novo mutations because they're robust and very accurate. We have 30x genomes giving you the coverage that you need to detect roughly, at least to what we believe now, detect all the genetically relevant variants in the clinical genetic setting. And we have come to a sort of situation, as also shown before, that we can scale up. We can automate certain steps. We can do 1,000 or more HiFi genomes on a single Revio. You've seen the outlook into the future that there might even be population-based scaling possible. So we have actually come from a place where we had 7% of patients being diagnosed for rare diseases, where we can relatively easily increase this to 50% if we want by just a single test. We believe that we can actually replace the vast majority, if not all, of our genetic testing by a long-read genome. Having shown you the studies that we have shown you today. So with that, I would like to thank a lot of people, but especially you for being here and listening to me, giving you this information. Thank you so much.